Hello everyone, I am Anjana Bhattacharya from Mirrors Bio, and I have the pleasure today of sharing with you some key transfection basics as part of this Transfection 101 talk. Nucleic acid delivery, or transfection as it is more commonly called, forms the basis of most routine cell biology experiments. Needless to say, therefore, understanding the basic mechanism of transfection and learning how to optimize it is critical to achieving effective experimental results. I hope what we discussed today as part of this 101 talk is going to help make your transfection experience easier and think about transfection in a new light. We are going to begin today's Transfection 101 with a brief introduction first about the Transfection Experts, the scientists at Mirrors Bio, followed by an insight into the basic mechanism of transfection. Once we've done that, we're going to discuss a few different methodologies that can be used to assess or evaluate transfection performance. This will nicely segue into our next topic of discussion that deals with establishing the ever more critical balance between gene expression and cellular toxicity following transfection. Lastly, but most importantly, we are going to elaborate on the key parameters that can be fine-tuned to maximize transfection output. So who are the transfection experts? We, the scientists at Mirrors Bio, take extreme pride in calling ourselves the transfection experts. The main reason for our pride being our singular and undiluted focus on transfection for the past 17 years. Our journey began in 1995 when three gene therapy scientists from University of Wisconsin-Madison, John Wolfe, James Hagstrom, and Vladimir Butker founded Mirrors. Since then, we have pioneered the creation of a wide range of transfection technologies using proteins, polymers, and lipids. Our key accomplishments include the introduction of the first ever serum-compatible transfection reagent for plasmid delivery, Transit LD1. This reagent finally allowed researchers to observe low toxicity and get physiologically relevant results during transfection. We were the first to also commercially introduce an siRNA transfection reagent, Transit TKO, and a serum-compatible reagent for large RNA delivery, Transit mRNA. We have simultaneously complemented transfection techniques with electroporation using our Ingenio electroporation solution for cells that are refractory to chemical transfection, and provided nucleic acid labeling technologies to track the intracellular traffic of transfection cargo. We are continuously innovating to address emerging transfection needs with the latest products in our portfolio, some of them being a high-performance animal-free reagent that is ideal for very hard-to-transfect cells, Transit 2020, a high-yield protein production reagent for suspension cells, Transit Pro Transfection Kit, and recently, biologically relevant transfection using the 3D transfection system. For us, transfection equals research, and I'm sure it does for a lot of you at the bench. A core of cell biology research depends on gene expression and knockdown techniques that are dependent on transfection. Also, there are novel applications such as high-throughput screening, therapeutic protein production, virus production, 3D transfection, and stem cell reprogramming that are dependent on how efficient the transfection method is. To give you an idea about how mainstay transfection is for biological research, I'm going to show you some figures in published research articles using the keyword transfection anywhere in their text that have been listed using the HighWire Press. This includes HighWire hosted journals plus PubMed listings. Since time immemorial, it is a whopping figure of more than 250,000 publications, of which close to 13,000 have been published in the year 2012 so far. So what does transfection mean? Interestingly, the term transfection finds its roots in virology, as it was first described as the infection of a cell with isolated viral nucleic acid, followed by production of the complete virus in the cell. We all know that today it has come to mean mostly the delivery of exogenous DNA into mammalian or insect cells. Not only does it apply to DNA, it also holds equally true for other nucleic acids such as siRNA miRNA, large mRNA, oligos, and even in some cases, proteins. There is a plethora of different transfection methodologies that are available for researchers currently. A majority of these are reagent-based methods that began with the use of calcium phosphate for condensing and complexing with DNA. However, this method, even though very, very cheap, suffers the caveat of being very, very irreproducible. A major class of current transfection reagents that you find in the market are based off of lipids. These are mostly cationic lipids that can interact with a negatively charged nucleic acid or liposomal formulations that can also effectively encapsulate the DNA. Another major component that is found in transfection reagents is cationic polymers. Some examples are polyethyleneamine, basic proteins or peptides, highly branched compounds called dendrimers, and other compounds such as DAE dextran. There are also instrument-based methods available for delivering nucleic acid inside the cell, the most important being electroporation that is highly used for cell types that are refractory to chemical transfection. There are also other instrument-based methods such as microinjection, biolistic delivery using the gene gun, and magnetic transfection using an externally applied magnetic field. Some researchers also refer to virus-mediated techniques as transfection. The reason that reagent-based methodologies are most popular among scientists lies in the simplicity of the transfection protocol using reagents. Here is a schematic to illustrate the simplicity of these transfection protocols. It is typically a 5-step quick protocol that involves first the dilution of the nucleic acid to be transfected in serum-free media. This is followed by the addition of the transfection reagent to the mix and let the complexes form between the transfection reagent and the nucleic acid with a simple incubation of 15 to 30 minutes. Following this, you simply add the transfection complexes directly to cells that are already grown in complete media. Depending on your experiment, you then incubate these cells for as long as required, anywhere between 24 to 72 hours, and harvest and perform your assay. Not only are these protocols very easy, these are also very amenable to high throughput. There's no instrumentation required except your routine cell culture supplies, and it affords decent transfection efficiency for routinely used cell types. The reason we need transfection reagents is because mammalian cell membranes are inherently very negatively charged. This causes a repulsion between the negatively charged nucleic acid that you intend to transfect and the negatively charged membrane. What the transfection reagents do in this case is, because most of them are made up of cationic components such as lipids or polymers, they can effectively neutralize the charge on the nucleic acid and also in some cases allow the condensation of DNA for effective uptake. Transfection is a very membrane-active phenomenon and a complex one at that. A lot of membrane biologists have been studying the mechanism of transfection for the past 20 years, but very little is understood about what exactly happens inside the cell. Here is a simple animation that shows what happens during transfection. When you take your already formed transfection complexes and incubate them with the cells, some of these transfection complexes go and bind to the cell membrane. These bound transfection complexes are then taken up by the cell using a variety of endocytic pathways. It is generally known that clathrin-dependent and caveolin-dependent endocytic pathways play a major role in the transfection complex uptake. There are also other pathways such as macropinocytosis and the rest that are involved in transfection. These endosomes then go through a variety of endocytic pathways and the transfection complex finally lands up in the really low pH environment of the endolysosomes. For effective functionality, however, the transfection complex and also the nucleic acid needs to escape this low pH environment and gets released into cytoplasm. For nucleic acids such as siRNA and mRNA, the cytoplasmic location is sufficient for functionality. However, in cases of plasmid DNA, there is further shuttling required into the nucleus so that gene expression can ensue. The reason transfection reagents are able to complex with nucleic acids effectively is inherent in their composition. A vast variety of commercially available transfection reagents are made up of cationic lipids. Cationic lipids are polar lipids that are formed of a highly positively charged head group attached to long hydrophobic tails. A few examples are DOTAP and DDAP. The polar nature of these lipids allows the positively charged head group to associate effectively with the negatively charged nucleic acid, while the hydrophobic tails can associate with themselves to form complexes called lipoplexes. These complexes do need to have a net positive charge to effectively associate with the cellular membrane that is negatively charged. In addition to cationic lipids, a few transfection reagents also contain another class of lipids called the helper lipids. A few classic examples of helper lipids are dope and cholesterol. The helper lipids, in conjunction with cationic lipids, form structures called liposomes that are effectively able to encapsulate DNA. These liposomes can be unilamellar, as seen in the middle here, or multilayered. The theme of a net positive charge with these liposomes continues, and it is often seen that these liposomal formulations tend to be more toxic than other transfection formulations. A very different class of components for transfection reagents than the already discussed lipids are cationic polymers. These compounds are very effective nucleic acid condensing agents because of their polymeric nature. An example is polyethyleneamine that can be linear or branched, as shown here. These polymers typically provide a lot of positively charged side groups, in this case amine groups, that can interact efficiently with the negative backbone of DNA and form complexes called polyplexes. These polyplexes also need to have a net positive charge. Now that we've discussed what major classes of compounds make up transfection reagents, I'm going to briefly describe to you what Mirror's transfection reagents are made up of. We employ unique lipid and polymer combinations that work synergistically to provide effective condensation and complexation of nucleic acids. Since our reagents are made up of both lipid and cationic polymers, the transfection complexes that result with the interaction of plasmid DNA are referred to as lipopolyplexes. Again, it is important for these lipopolyplexes to have a net positive charge. Throughout the years, we have seen that these lipoplexic formulations tend to be the least toxic formulation compared to all other classes of transfection formulations. Now that we have seen how important the chemical composition and the type of complexes can be in deciding how efficient or toxic the transfection reagent is, there are also other features of transfection complexes that need to be considered for efficient transfection. One such parameter is the size of transfection complexes. It can range from anywhere between 40 to 1,000 nanometers. With our reagents, we have typically seen this to fall in the range of 300 to 500 nanometers. Keep in mind that either very small transfection complexes or very large transfection complexes are not very efficient at uptake by the cell. Another key parameter in the grand scheme of transfection is the overall charge of the transfection complexes. As I mentioned in the previous few slides, all kinds of transfection complexes, either be lipid or polymer based, should have a net cationic charge. Now this really needs to manifest in terms of an overall positive surface charge density of the transfection complexes. This surface charge density by gene delivery scientists is calculated in terms of the zeta potential, which typically should be on the slightly positive end. Another parameter that is related to charge is the charge ratio of the transfection complexes. This charge ratio essentially is the molar ratio of the positively charged moieties on a transfection reagent that complex with the negatively charged phosphate moieties of the DNA backbone. This is often referred to as by gene delivery specialists as NP charge ratio or just NP ratio. Here is an example of a branch PEI molecule that offers several amine groups to complex with the backbone of DNA. So in this case, the charge ratio effectively would be the molar ratio of all the amine groups that are available to access the phosphate backbone of DNA to the phosphate groups on the DNA. This charge ratio is indeed a very important parameter. You'll realize this because for a lot of transfection optimization, pretty much with every transfection reagent, when you're asked to optimize the reagent to nucleic acid ratio, what you're essentially optimizing is the charge ratio of your transfection complexes. Here is an example of different reagents, reagent A, reagent B, and reagent C, that are being optimized for different ratios of the reagent to a fixed amount of nucleic acid in this case. And as you can see, different transfection reagents, because of their inherently different nature, show different profiles in terms of what charge ratio works the best for transfection. So keep in mind that this charge ratio indeed does impact your transfection experiments directly. A key aspect of understanding transfection also involves figuring out how to evaluate transfection performance the best. Obviously, if we need to understand a process well, we need to also understand how to assess it well. Most researchers throughout the world rely on the use of reporter systems for evaluating transfection performance. The most commonly used reporter systems include green fluorescent protein, which can easily be qualitatively assessed by simply looking at your cells under the microscope and figuring out which ones are fluorescent versus not. You can also do much more comprehensive quantitative analysis if you choose to assay the green fluorescent protein expression in a flow cytometer. Another very popularly used reporter system is the luciferase reporter system. The reason for its popularity is because it's a highly sensitive reporter assay with a very broad dynamic range. We routinely use it in our labs because it gives us the most sensitive measure of comparing transfection performance between different formulations, either ours or competitors. A less commonly used reporter system that is used by labs is the beta-galactosidase assay system. Again, it can be qualitatively assessed by simply staining your cells blue using a colorimetric substrate called XGAL, or you can also do quantitative measurements using a variety of chemiluminescent and colorimetric substrates that are available for beta-galactosidase enzyme. A unique reporter system that lends itself very useful in cases where you have to look for a secreted reporter molecule is the secreted alkaline phosphatase or SEEP. This method is especially useful in cases where you have to do large batch cultures where secreted proteins can be assayed, for example, antibodies, growth factors, etc. Another advantage with this method is because the protein is secreted, you really don't need to harvest the cells in order to assay the transfection output. So far, we have talked about indirectly assessing transfection output because we are mostly employing reporter systems that assay the indirect expression of the transfected nucleic acid. But how about visualizing nucleic acid delivery directly? This can be done easily by using technologies such as our label-it reagents that are covalent linkers that attach to the DNA molecule with either fluorescent moieties such as cydies, fluorescein, rhodamine, etc., or non-fluorescent tags such as biotin and DNP. The labeling protocol is a very simple protocol where you take your transfected plasmid or any other nucleic acid, you simply add the label-it reagent to it and incubate for 1 hour at 37 degrees Celsius. During this reaction, the label-it linkers attach covalently to the backbone of the nucleic acid that is incubated with the reagent. There are also methods available to purify this labeled nucleic acid from the unreactive components of the labeling reaction, following which we are left with labeled plasmids or any other nucleic acid such as siRNA or mRNA that can be used for your transfection experiments. An important aspect of labeling nucleic acid for tracking purposes is to make sure that they retain their functionality after labeling. It is a generally known problem with labeling techniques that very high labeling efficiencies can either cause the quenching in case of fluorescently labeled nucleic acids because the fluorophores are too close together, and it can also affect functionality. For example, a case with plasmid DNA is that gene expression from very, very highly labeled plasmid DNA templates could be hampered. This could either affect transcription or translation of the mRNA. Also, in case of siRNA, a concern is that knockdown could be affected because of the labeling of the siRNA. This is because hybridization of the siRNA might be affected. To alleviate this concern, we have done a lot of optimization with our labeled technology, where we have optimized the number of labels that can be on a plasmid or an siRNA molecule and yet retain functionality. Here is a chart illustrating just this point. As you can see here towards the left, the fewer the number of labels per plasmid, in the range of 5 to 10, the higher the luciferase expression with a wide variety of labeled plasmids, either with rhodamine, Si3, biotin, etc. Using our labeled technology, we have optimized labeling densities very, very finely to make sure that labeled nucleic acid maintains or retains function for a variety of applications. The first example here is with Cas7 cells that have been transfected using a rhodamine labeled DNA expressing nuclear YFP. As you can see here in the image panel, at 1 hour post-transfection, you begin to see puncta that represent complexes of this rhodamine labeled DNA. At 4 hours post-transfection, this labeled DNA starts expressing nuclear YFP, and the expression increases at later time points of 8 hours and 24 hours. Therefore, it is clearly evident that in this case, with the labeled DNA, there is absolutely no hampering of expression, implying that there is no problems with gene transcription or translation. Furthermore, in cases of siRNA, we have demonstrated that labeling densities afforded by our kits do not affect knockdown. Here is an example of HeLa cells that have been transfected using fluorescein-labeled siRNA. And as you can see here, you can visualize the fluorescein-labeled siRNA, the green dots over here, and also see that compared to an unlabeled control in the chart below, neither of the labeled siRNAs are significantly different in terms of knockdown. They actually retain the same level of knockdown as the unlabeled siRNA. So this tool really provides us with a very carefully optimized system for nucleic acid tracking. If you recall from the earlier mechanistic slides about transfection, it is a highly membrane active phenomenon. There's a lot of endocytic pathways involved, and there's a lot of membrane reorganization within the cell following transfection. A harsh reality, therefore, of most transfection methods is the associated cellular toxicity incurred by these techniques. Here is an image illustrating how transfection can really affect cell health. Typically, you would want your cells to look healthy and morphologically fine, have no effects on cell viability, etc. following transfection. These are HeLa cells that have been cultured and transfected using several different transfection reagents. A lot of different labs use HeLa cells as their cellular model. Unfortunately, oftentimes, depending on the transfection reagent that you used, your cells will end up looking like this. Now let's look closely at these HeLa cells. Depending on what transfection reagent you use to transfect these cells, here are a few examples. The first on the left is our Transit LT1 reagent, followed by Transit 2020, another high-performance reagent by Mirus. And in comparison is a liposomal formulation that is popularly used called lipofectamine 2000. At the 8-hour time point, you barely notice any differences in cell morphology with either of these reagents. The cells look just fine. However, as you progress into the 24-hour time point post-transfection, the lipofectamine 2000 formulation wreaks havoc on the cells. The cells are all shriveled up and rounded compared to the healthy-looking cells with the Transit LT1 and Transit 2020 formulations. This also correlates with the GFP expression profile because the cells that are rounded up and shriveled, even though they might be expressing some GFP, are essentially lost during transfection. This drives home the point that striking the balance between expression and toxicity is really critical for your transfection experiments. Apart from just visually assessing cell health, there are a lot of very sensitive assays available to detect cellular toxicity. One that we regularly employ in our labs is the lactate dehydrogenase assay, or LDH assay. Lactate dehydrogenase is an enzyme that is expressed by all cell types. What this assay does is assesses the membrane leakiness by assaying the amount of LDH release into the medium from the cells. So indirectly, it assesses the level of membrane damage because you have an otherwise cytosolically located enzyme that is now getting released into the supernatant because the cells are leaky. Here you can see a data set of HeLa cells that have been transfected with a luciferase encoding plasmid. This data shows expression and also the corresponding toxicity values in terms of the LDH release from the matching cell supernatants. Again, as you can see here, with lipopoliplexic formulations such as Transit LT1 and Transit 2020, you see good expression in these HeLa cells and correspondingly low toxicity. However, with liposomal formulations such as lipofectamine 2000, the expression of luciferase is twofold lower, at least, and also the levels of toxicity are very high, close to 60% compared to less than 10% with the LT1 and 2020 formulations. With these gross effects on morphology, viability, and membrane leakiness, one wonders what really happens inside the cell. What causes the cells to look so sick or vice versa? We looked into this further by looking at the gene expression profiles by using an RT-PCR-based method to analyze the expression of 84 key genes involved in cytotoxicity. This experiment was done with HeLa cells that was transfected using either Transit LT1, Transit 2020, or lipofectamine 2000. In this experiment, a non-coding plasmid was used to make sure that there were no gene expression associated changes on the overall cytotoxicity profile. As you can see here, cells hugely alter their gene expression profile following transfection. Of the 84 key genes that were looked at, the alteration in gene expression was maximum with liposomal formulations such as lipofectamine 2000. Expression of more than 9 and more than 25 transcripts were altered by lipofectamine 2000 transfection at 8 and 24 hours respectively. Compared to this, very few genes, only 3 and 9 respectively at the 8-hour and 24-hour time point, were affected by cells that were transfected using the Transit LT1 and Transit 2020 complexes. You can get a further idea of the magnitude of the gene expression alteration by looking at these results in the form of a Venn diagram. As you can see here, many more genes are upregulated or deregulated with lipofectamine 2000 at either the 8-hour time point or the 24-hour time point. Could these gene expression changes be affecting the pathways that you're studying? We conducted pathway analysis using the Ingenuity software on these RT-PCR results, And we found that the most overrepresentative pathways that were affected by transfection were associated with oxidative stress response and xenobiotic metabolism. Among the genes involved in oxidative stress response, that is shown here in the topmost panel, the DDIT3 mRNA expression was increased approximately 50-fold in cells that were transfected with lipofectamine 2000, whereas it was significantly lower with cells that have been transfected with either Transit LT1 or Transit 2020. Based on this pathway analysis, we also found that the most significant biological function affected in response to transfecting cells with lipofectamine 2000 is related to unfold protein and DNA damage response. This pathway includes deregulated genes coding for heat shock proteins such as HSPA5, HSPA1A, DNAJ, subfamily B, CDKN1A, and tumor protein 53. What was concerning to us and could be to you as well is that a lot of people are actually doing the research on these particular pathways. So if you use transfection reagents that could significantly upregulate or deregulate the gene expression profiles of any of these proteins that you're looking at, it would be a major concern for your experimental output. Furthermore, it is seen that different cell types show different expression toxicity profiles than, say, HeLa cells. Here are a few examples of HepG2 cells, THP1 cells, and T2 cell and HUVEC cells. As you can see here, you do get a good balance of expression versus toxicity that is measured here in terms of luciferase expression versus LDH release with either Transit LT1 or Transit 2020 in most cases. With lipofectamine 2000, you can get moderate levels of expression, but the toxicity levels as measured via the LDH assays are exceedingly high. So this further emphasizes the point that it is very, very critical to choose the right transfection reagent for your particular cell type. With most cell types, you can optimize your transfection even further to get better results. A major reason for optimizing transfection lies in the current cell biology research trends. So far, most researchers have been using well-established cell lines that are relatively easier to transfect. However, more and more research is trending towards harder to transfect cell types, such as primary cells and embryonic stem cell products. Furthermore, we know that not every cell type behaves similarly with every transfection reagent. That makes it more imperative to optimize your transfection conditions to get that additional transfection output from your experiments. Improving transfection performance is fortunately not very hard to do. The answer lies in the simple transfection workflow if you just look at it sequentially. You begin with looking at your serum-free media for complex formation, the nucleic acid that has to be transfected, the complex formation time, cell culture conditions such as confluency or suspension cell density, and in the end, the harvesting time for your assay. The serum-free media that you use for your transfection complex formation is one of the most critical factors in transfection. The reason serum-free media is recommended for preparing pretty much all the transfection complexes across different transfection reagents is because serum is known to contain nucleases that are likely going to chew up your nucleic acid substrates even before it sees a transfection reagent. So it is an absolute no-no to use serum-containing media for complex formation. Another major factor that could disturb transfection is antibiotics. A lot of antibiotics, such as kanamycin, can be highly charged and can disrupt transfection complex formation. On the same theme, other highly charged compounds, for example, polyanions such as heparin sulfate, can disrupt transfection complex formation. We also know that in certain applications, for example, therapeutic protein production, where suspension cells are cultured in large bioreactors and are maintained in the presence of surfactants such as pluronic F68, This compound can significantly obliterate transfection efficiencies. Therefore, all of these categories of compounds or highly charged molecules should be completely avoided at the step of transfection complex formation. If needed, some of these compounds can be added later on during your transfection incubation. To further emphasize how much serum-free media can affect transfection, here is some data with GFP transfection of 3D3 cells with complexes made using various different serum-free media. As you can see here, serum-free media such as Optimum, DMEM, and RPMI 1640 afford decent levels of transfection efficiencies in this case. However, I would like to particularly note that certain media such as 293-SFM2, the HiQ series, the XL293 media, and the HiQ CDM CHO media completely obliterate transfection. One major cause for this inhibition of transfection could be that these media are known to be used for suspension cell culture, in which case they might have surfactants like proloric F68 that are completely inhibiting transfection in this case. In specialized applications such as large-scale protein production, the overall transfection and therefore the protein yields can also be affected by the complete media that the cells are grown in. This is because in these applications, the final cell density is very, very important. Here is some data shown with our Transit Pro transfection kit in which an antibody-expressing plasmid is transfected into CHO suspension cells that have been adapted to a variety of different media. As you can see here, you get maximum expression in the case of CHO cells that are adapted to the freestyle CHO expression media, followed by the other media types. So in such cases, where the final cell densities might be very important, it is highly critical to optimize even the complete media choice. The next critical parameter in the transfection workflow is the nucleic acid that has to be transfected. The most important attribute of the transfected nucleic acid is the level of its purity. For example, in the case of DNA transfection, it is critical that contaminants such as protein, carbohydrate, and lipids should not be present in the DNA prep as they may affect transfection efficiency. Most maxi prep kits that we use for DNA preparation in the labs do account for this purity level. We should also make sure that the ideal plasmid preparation exhibits an absorbance ratio of 260 by 280 of greater than 1.8 and ideally lower than 2. Like I mentioned, most maxi prep kits allow us to get relatively pure levels of DNA. However, the biggest culprit when it comes to transfection is the endotoxin contamination of the DNA prep itself. This endotoxin is carried over from the E. coli cells because it is a part of the outer membrane of gram-negative E. coli. The endotoxin molecules are known to be very, very toxic to mammalian and insect cells. Here is some data illustrating the toxicity of endotoxins with respect to transfection. As you can see here with the commercial plasmid in which the endotoxin levels have been removed to very, very low amounts, you get good amounts of luciferase expression. However, with the crude plasmid that has exceedingly high levels of toxin, the transfection is completely inhibited. Now, when you take this crude plasmid and purify it and remove the endotoxin with 3 rounds of treatment with our Miraclean endotoxin removal kit, the levels of expression of luciferase are retained back to the same levels as found with the commercially purified plasmid. Therefore, it is very, very important to remove all traces of endotoxins from your plasmid preps for the best transfection. This is particularly more critical in sensitive cell types, for example, cell types of immunological lineage. The features of the nucleic acid that has to be transfected also plays a critical role in transfection. For routinely transfected nucleic acids, such as plasmid DNA, we all use common parameters such as CMV promoters, etc., that take care of maximal expression. However, in cases of substrates such as mRNA that are particularly sensitive, it is very important to consider the various features of the mRNA for maximizing transfection. mRNA can be easily produced in labs using an in vitro transcription reaction. However, the mRNA produced using in vitro transcription does not have the naturally occurring 5' cap structure, nor does it have the polyadenylated tail that helps with the mRNA stability and processing. There are a lot of commercially available kits now that let you add a cap analog on the 5' end or an iris that substitutes for natural capping. And also, polyadenylated tails can be added by using tailing kits that contain poly-A polymerase. Here is an example of how mRNA tailing can significantly improve expression. This experiment shows the expression of a luciferase transcript that is tailed and also another version that is non-tailed in raw 264.7 cells. The different colored bars represents the different times of harvest. And as you can clearly see here, at earlier time points such as 14 hours or even 24 hours, there is much better expression of the luciferase transcript when it is tailed versus non-tailed. So these small features in the nucleic acid can contribute to a great extent in maximizing your experimental results. Needless to say, the nucleic acid dosage also matters. We have seen routinely that doubling the dosage of DNA in harder to transfect cell types such as Jurkat cells or raw 264.7 actually improves the levels of expression quite a lot. Here is an example of how DNA dosage increases expression. In the red bars here is the ratio of 3 microliter reagent to 1 microgram DNA for 3 different cell types, COS7, CHOK1, and HEK293s. The gray bars represent the same ratio of the reagent to nucleic acid, but here the amount of nucleic acid is threefold lower, almost threefold lower, 0.4 microgram DNA. As you can see here, there is a corresponding decrease in the level of expression with the lower dosage of DNA. This dosage theme continues through other nucleic acid substrates such as mRNA. Here's an example of DC 2.4 cells that have been transfected with increasing dosage of a GFP mRNA from 0.5 micrograms to 2.5 micrograms. And as you can see here, not only is a larger percentage of cells expressing GFP with the higher dosage of the mRNA, they're also brighter. The same holds true for siRNA optimization. In case of siRNA, it's even more critical because higher amounts of siRNA can lead to non-desired off-target effects. As shown in the chart here, you can reduce the amount of siRNA to as low as 1 nanomolar to get effective knockdown. This also helps with any fears about off-target effects affecting your experiments. There are a few specialized applications that are affected with nucleic acid dosage. One such application is if you're overexpressing toxic proteins. The problem here lies in the fact that the overexpression of the toxic protein itself is going to kill the cells. In this case, a neat trick that you can employ is actually lower the plasmid dosage for the toxic protein. So for example, if you were transfecting 1 microgram of the toxic protein encoding DNA, you can lower it down to 0.1 microgram, that is 10-fold less, and add remaining non-competing cloning vectors such as a PUC18 or a 19 vector. Here is some data showing the expression of luciferase from a co-transfection of PUC18 and PCI-LUC. As you can see here, as you increase the amount of the non-completing cloning vector, the amount of luciferase expression decreases. In case of toxic proteins, this would be deliberate but would allow your experiments to succeed. The same holds for eGFP expression in this experiment, where you titrate down the amount of GFP to 0.25 micrograms from 1. And you can still see some GFP expression without having to hamper your experimental results. There are also different co-transfection scenarios that are routinely employed in the labs. One very popularly used co-transfection experiment is actually virus production. In this case, researchers typically take anywhere between 3 to 17 or even more viral plasmids and transfect cells together with them. Following transfection, the viable virus is produced and released outside of these cells in the supernatant. In most cases, people co-transfect multiple plasmid DNAs, but they can also co-transfect other nucleic acids such as multiple siRNAs, oligos, or mRNAs. It is very critical to always pre-mix multiple nucleic acid to prevent preferential complexation with the transfection reagent. If you have a mixture of 3 plasmids, always pre-mix them to avoid complexing one plasmid better than the other. Also, depending on your application, it is critical to choose the molar ratio of the multiple plasmids carefully. In cases of some reporter systems, only a very little amount of the reporter plasmid is added to the transfection mixture, whereas cases where you want to get equivalent expression from 2 plasmids, you would want to ideally add an equimolar ratio. There are also co-transfection scenarios where 2 different nucleic acids need to be co-transfected. Some examples are plasmid DNA and siRNA co-transfection, plasmid DNA oligo, or plasmid DNA and mRNA co-transfection. Of these, plasmid DNA and siRNA co-transfections are actually done quite routinely. One of our reagents, the Transit TKO reagent, works very well for this application. However, there are some points to consider when you're transfecting different nucleic acids. You should ideally form the complexes with each respective nucleic acid separately, again to avoid preferential complexation of one nucleic acid over the other. You can form these complexes separately and then just simply mix the complexes before adding to the cells. The amount of time that you let the complex form is also very critical in the transfection workflow and can be optimized in some cases. Most transfection protocols call for an incubation time of 15 to 30 minutes for transfection complex formation. During this time, the transfection reagent complexes effectively with the nucleic acid and also allows the right size of complex to form. The problem with letting it go too little is that you don't have the right size of the transfection complexes formed, and the problems with letting it exceed too long is that the transfection complexes become too big and are not able to be engulfed by the cell anymore. Here is an example of transfection complex formation time with plasmid DNA in CHOK1 cells. These cells have been transfected with a plasmid that expresses luciferase and is complexed with the Transit LT1 reagent. As you can see here, anywhere between 15 to almost 50 minutes is a big window for the plasmid complex formation. Sometimes, though, we can see a precipitous decrease after 30 to 40 minutes, and generally we do not advise going further than 30 minutes of incubation for transfection complexes with DNA. This transfection complex formation time is even more critical with sensitive substrates such as mRNA. As you can see here in this chart, typically anywhere between only 5 to 10 minutes is required for effective transfection complex formation, in this case with CHOK1 cells that have been transfected using a luciferase mRNA. We do not advise going any further than 10 minutes in case of mRNA expression. Cell culture conditions such as cell confluency and density also play a very important role in transfection. In case of adherent cells, the overall cell confluency can have a huge impact on transfection. Here is an image of COS7 cells showing different levels of confluency from 25% to 90%. Cells that are very low confluency, for example 25%, generally are not very good in terms of overall cell health and therefore do not fare well with transfection. A typically good range for transfection is 50 to 70% cell confluency. On the other hand, very high cell confluency, such as 90%, lead to contact inhibition of the cells and prevention of cell division. In this case, the plasmid DNA is unable to be uptaken by the nucleus because of the dissolution of nuclear lamina that occurs in dividing cells, and that really hampers transfection efficiencies. To illustrate this point, here is a chart of 3D tissue cells that have been transfected with a luciferase encoding plasmid at 75% confluency versus 100% confluency. As you can see here, because there is active cell division at 75% confluency, there is higher amounts of luciferase expression. However, when the cells are 100% confluent, they are really contact inhibited, and there is no room for cell division whatsoever. This further translates in terms of lower luciferase expression. In case of applications where suspension cells are cultured, the overall suspension cell density also impacts transfection, much like adherent cell confluency. The applications in which this is affected are large-scale protein production applications, in which the optimal cell density typically tends to be anywhere between 0.5 to 1 million cells per mil, as shown in this chart below, of chose suspension cells that are transfected with an EGFP plasmid. At lower cell densities, there is not enough cell mass, whereas at higher cell densities, there again probably exhaustion of cell resources, which prevents the cells to actually transfect very well. Another very neglected issue related to cell culture is mycoplasma contamination. This can be very hard to detect because your cells look just the same as routinely cultured cells. They do not show any visible signs of contamination. That is not to say, though, that mycoplasma contamination does have a huge impact on transfection. It can lead to highly altered transfection results. In some cases, it leads to a severe decrease in transfection performance, whereas in some cases, you might get unrealistically high transfection rates. With a lot of PCR kits available now to detect mycoplasma, it has become easy to actually perform mycoplasma contamination tests in the lab. Our advice, if you suspect mycoplasma contamination, is to completely discard contaminated cultures. Or if you do not have that option with precious samples, you can always treat with antibiotics that are recommended for mycoplasma elimination. The last parameter in the transfection workflow that can be optimized is the harvesting time for your experiment. The harvesting times for different experiments are determined by the experimental goals themselves. For example, typical harvesting times for gene overexpression studies can range from anywhere between 24 to 72 hours depending upon the protein that is to be expressed. For gene knockdown, it is usually recommended to wait slightly longer, from 48 to 72 hours, to make sure that any residual protein expression that could contribute to misinterpretation of knockdown results is completely taken care of. For mRNA translation, which is susceptible to stability issues, earlier time points are desirable, for example, anywhere between 4 to 24 hours. Again, it might also depend on the mRNA itself. For specialized applications such as protein production, a longer incubation time is recommended, anywhere between 2 to 14 days, to increase the cellular biomass and therefore the associated protein yield. Here is an example of a human antibody that is produced using our Transit Co-transfection kit and also competing reagents such as linear PEI and Freestyle Max. As you can see here, in all three cases, there is an accruing of the antibody titers with increasing incubation time. So in this case, it is highly beneficial to actually let the harvesting times to be much longer. The harvesting times are also affected by issues related to nucleic acid turnover. Here is an example of Firefly luciferase mRNA that is delivered to NIH-3 cells using our Transit mRNA kit. As you can see here, possibly related to the issues of mRNA stability and turnover, earlier harvesting time points, such as a 12-hour time point post-transfection, is ideal for harvesting in this case. Before and after this time point, there is a rapid decrease in the level of expression of luciferase. This concludes our transfection optimization section of today's 101 talk. However, I would like to emphasize one last point when you're comparing transfection performance between different transfection reagents. Transfection results can vary quite a lot day to day based on the parameters that we discussed just in the previous slides. Therefore, it is very, very important that if you're doing any comparison of transfection reagents, that you perform the reagent comparison on the same day with the exact same batch of cells. It is also very important to follow reagent-specific protocols with each transfection reagent. Here's some data exemplifying this. In the first chart, you can see that reagent A outperforms reagent B if you compare day 1 transfections against day 2 transfections. However, once you have data from both day 1 and day 2, you can see that it's simply a matter of cell health in this case. It just turns out that the cells were transfecting much better on day 1. But they were transfecting much better with reagent B. So once you have the complete picture, you can see that in this case, reagent B outperforms reagent A. However, if you were comparing day 1 to day 2, that would not be the case. This brings us to the end of today's Transfection 101 talk. I hope that each of these modules that we covered in today's talk has helped you somewhat in gaining transfection knowledge. If you'd like to see any of these modules separately or the complete 101 talk, you can simply visit our YouTube channel and watch these videos over there. You can also learn more on our website, www.thetransfectionexperts.com. We have a technical resources section that you can find useful for gaining more information about transfection. We have tips from the bench there to help with particular experiment types such as DNA transfection or siRNA transfection. We also have a comprehensive transfection database called the Reagent Agent, which gives you recommendations for your particular cell type and nucleic acid combinations. You can also speak with the Transfection Experts directly by either calling us or emailing us at our tech support address. Thank you so much for watching and listening to this tutorial. If you have any feedback to give to us or any questions, please do contact us.